19.4, formed elements. We're going to talk about all the various formed elements that we'll find within blood. Um, Red blood cells, you'll hear them referred to as RBCs or erythrocytes. This is 95% of the volume of formed elements, whereas white blood cells, known as leukocytes or WBCs, are going to form the other 5% along with platelets. When we talk about white blood cells, we're going to have two different types. We're going to have granulocytes, which is where we have a large cytoplasm that's going to have these large granules. Typically, we'll have multi-lobe nuclei, and we have three distinctive types: neutrophils, eosinophils, and basophils. And then we have agranulocytes, where the cytoplasm is going to contain really small granules, and typically nuclei that are not lobed. Beware that we are looking at a 3D object, typically in a 2D plane. So it can be a little hard to denote the nuclei and what type of white blood cell it is. But when we get into having to identify the different types of white blood cells, we'll have multiple tips so that you can identify each readily, not just based on whether they're granulocytes or agranulocytes. But we have two distinctive types of agranulocytes, and those are going to be lymphocytes and monocytes. Platelets, which I'm sure most of you have heard of, and I referenced in a previous video, we technically refer to them as thrombocytes, and these are cell fragments. That's when we talked about how formed elements are all the cells in the cell fragments. Platelets are the cell fragments. Platelets help form the platelet plugs. They're going to release various chemicals that are necessary for blood clotting, and they're extremely crucial for preventing excessive hemorrhaging and losing large amounts of blood. So here's just an image of some formed elements. You can see here that we have our red blood cell here, we have a white blood cell here, and we have our cell fragment, which is our platelets here. One thing I do want to point out in this image is looking at the shape of the red blood cell, and we'll get into this a little bit later, but the red blood cell has a biconcave disc shape. It kind of looks like a Cheerio, not with a necessarily hole in the middle, but a divot or a little cave in the middle, and we'll see why that's relevant and later on in this lecture. So talking through all the different formed elements, we'll start with red blood cells and just give you a little bit more description of what they are, the functions, and the relative abundance. So red blood cells, biconcave disc, and there's no nucleus here. We'll see that when we go through the process of forming red blood cells, that they will lose the nucleus before they become mature. Contains hemoglobin. That's what's going to give it its red color. And its main function is to transport oxygen and carbon dioxide. A lot of people don't necessarily realize that we all think of red blood cells as transporting oxygen, but we forget about the carbon dioxide. So it is important to remember that it is transporting both. Something that's also extremely useful when looking at imaging with red blood cells in it is that we refer to red blood cells as a histological ruler. What that means is that a red blood cell is typically 7.5 microns in diameter, and that does not have a lot of variability between people. Almost everyone's red blood cells are 7.5 microns. So when you have a red blood cell in an image, you can compare the size of that red blood cell to adjacent tissues or objects, and you can get a general idea of the size of things. So typical abundance, and this is going to be the number of cells per microliter. So 4.2 to 5.4 million in females and 4.7 to 6.1 million in males. Now, it's not too often that I require you to remember strict values, and this is one of those cases where you don't have to necessarily remember that females are 4.2 to 5.4 million. All I'm trying to express is that you recognize the difference in abundance between females and males, not remembering specific values. So white blood cells, these are typically spherical cells, and they have a nucleus. We have 5 different types of white blood cells, which we referenced earlier, and each has a specific function that we will go over later. Typically 4,500 to 11,000 per microliter. So we're going to look at granulocytes first and their functions and what percentage of blood that they're of the white blood cells that they're making up. So we'll start with the neutrophil. Nucleus of a neutrophil is typically 2 to 5 lobes. Lots of cytoplasmic granules. It stains light pink or reddish purple, and it's going to be 10 to 12 microns in diameter. And its main function is that it's going to destroy microorganisms and other substances through phagocytosis. Neutrophils are Usually the first ones to get to a site of infection, and they make up a majority of the white blood cells. They're between 55 to 70% of the white blood cells. Now, these are values that I do care that you know, but I want you to know which white blood cells in order have the most abundance. So neutrophils will be the most abundant type of white blood cell. Now, eosinophils are extremely interesting. Typically are going to have a bilobed nucleus, extremely granular looking, really orange or red stained, and they function in attacking parasitic worms. It can release various chemicals that will modulate inflammation. One caveat to eosinophils is that they can have a poor impact on airways during asthma attacks, can increase inflammation. And these will be 1 to 4% of your total white blood cells. So the last granulocyte is the basophil. This will have a nucleus with 2 indistinct lobes, very hard to distinguish a basophil sometimes. You'll have the cytoplasmic granules, typically will stain a darker purplish blue color, and they function in releasing histamine. This is going to help promote inflammation, and they can also prevent blood clotting by the use of heparin. You may have heard of people being on heparin drips, and that is just a chemical that is used as a blood thinner. These are going to contribute to about 0.5 to 1% of total white blood cells. So now on to our agranulocytes, the lymphocyte and the monocyte. Lymphocytes typically have a round nucleus. Cytoplasm around the nucleus is extremely thin. Produces antibodies and other chemicals responsible for destroying microorganisms. Plays a role in graft rejections if you have skin grafts. Tumor control, regulation of the immune system, and also is a large contributor to allergic reactions, anaphylactic shock, and things of that in that domain. 20 to 40% of white blood cells are going to be lymphocytes. Monocytes, nucleus is round, kidney-shaped, can be horseshoe-shaped. I think monocytes typically are harder to distinguish because you can't necessarily tell off the nucleus, but they are significantly larger in size, which does help pinpoint a monocyte. Can go through phagocytosis, cells in the bloodstream, can leave the blood and become a macrophage, which can phagocytize bacteria, dead cells, cell fragments, and other debris within tissue. And these are going to contribute 2 to 8% of all white blood cells. Platelets are just the cell fragments that we talked about. They're going to play a role in releasing chemicals to help with blood clotting. They're going to form the actual platelet plugs within blood so that we can limit hemorrhaging. So we talked about all these formed elements, but we need to talk about how did we create these various formed elements. And this is through a process known as hematopoiesis or hemopoiesis. And this is the process of blood cell production. It varies throughout the lifespan. So in infants, this is going to occur in the yolk sac of the embryo, which you probably are not familiar with, but we'll talk about it during our development chapter at the end of the semester. Can also occur in the spleen, the liver, the thymus, red bone marrow, and the lymph nodes. Once birth occurs, red bone marrow and lymphatic tissue is where this will occur. Red bone marrow in the adults, which we'll specifically find in the ribs, the sternum, the vertebrae, the pelvis, proximal humerus, proximal femur. But to form all of our formed elements, we need to start with a stem cell. And the stem cell is a cell that has the ability to differentiate into various cell types. So all formed elements are going to be derived from a single stem cell known as a hemocytoblast. So when a hemocytoblast divides, it will divide into 2 daughter cells. One of those daughter cells will remain a hemocytoblast for future divisions, while the other is then able to differentiate into either a lymphoid stem cell or a myeloid stem cell. Myeloid stem cells are going to become proerythroblasts, which will develop into red blood cells, monoblasts, which will develop into monocytes, myeloblasts, which will develop into basophils, neutrophils, and eosinophils, or megakaryoblast, which will develop into platelets. Lymphoid stem cells will become lymphoblasts, which will then develop into lymphocytes. Now, this image might be a little hard to see, but I would zoom in and blow it up on your computer. But this is a good way to remember the lineage of how we're creating all these various formed elements. So we have our hemocytoblast here, which can then differentiate into a myeloid stem cell or a lymphoid stem cell. Remember, now these are not showing the two cells that divided from this. One of the cells of the division will stay a hemocytoblast. This is just showing the pathway that the one cell can go through. It can either become a myeloid stem cell or a lymphoid stem cell. If it becomes a lymphoid stem cell, it can turn into a lymphoblast, which will then form a lymphocyte. The other option is that we can go from a myeloid stem cell into a proerythroblast, to an early erythroblast, intermediate erythroblast, to a late erythroblast. And the key here is that this is when the nucleus is extruded from the cell, which will then turn it into a reticulocyte, which will then form our mature red blood cell. Now, the other option that we have is that the myeloid stem cell can then turn into a megakaryoblast, which will form into a megakaryocyte. And this megakaryocyte will then be broken up into individual little pieces, which end up forming our platelets. Now, myeloid stem cell has another option. It can go into a myeloblast. It can go into a progranulocyte, which will have our basophilic myelocyte, eosinophilic myelocyte, and neutrophilic myelocyte, which will then turn into those representative band cells, which will then turn into our basophil, eosinophil, and neutrophil. Now, if the myeloid stem cell goes into a monoblast, differentiates into a monoblast, that will then form a monocyte. The process of hematopoiesis seems daunting, but it's extremely important to recognize the easy portions of it and then kind of work yourself backwards. So hemocytoblast into a lymphoid stem cell, a lymphoblast, which will form a lymphocyte. That trajectory makes sense, not that many steps. Same thing with the monoblast or the myeloid stem cell going to the monoblast, that will form the monocyte. So once you have that part down, exclude that and then really hone in here. And we'll talk about this a lot in class. This is pretty important for creating our formed elements, which are obviously necessary for blood to function appropriately. So the production of formed elements is regulated by hormones and chemicals. So chemical signals such as colony stimulating factor, that's going to help regulate development. You'll see this referred to as CSFs within the textbook. That's probably how I'll refer to it most frequently. And then erythropoietin, this is the big one, EPO. This is a hormone released by the kidneys that will actually stimulate red blood cell production and development. So I alluded to this earlier, red blood cells or RBCs. They have a biconcave disc shape. They're 7.5 microns in diameter. No nucleus in a circulating red blood cell. It's already been extruded. One thing that makes red blood cells special is that they have really amazing capabilities that allow them to squeeze through really tight places like capillary beds, the smallest of our circulatory system. So they're flexible. They're able to bend and fold without destroying their structural integrity. That gives them the ability to pass through small vessels and move within the flow of blood. Some capillaries are just a micron or 0.1 microns larger than a red blood cell. So red blood cells can be lined up in single file line passing through a capillary bed. So it's extremely important that they have the ability to bend and move so that they don't get stuck in capillary beds and cause clots. Red blood cells also are going to contain hemoglobin, ATP, lipids, and carbonic anhydrase, which we'll talk about a little bit later. But you can see in the image below our biconcave shape. This is looking top down into it. And this is a side view where you can really see the divot in the center. So functions of red blood cells, the main one is to transport oxygen from the lungs to the tissues. 98.5% is going to be attached to hemoglobin. 1.5% of oxygen is going to be dissolved within the plasma of blood. So majority of our oxygen is getting to tissues attached to the hemoglobin found on a red blood cell. Carbon dioxide can be transported from tissues to the lungs in three different ways. So 7% will be dissolved in plasma, 23% in a combination with hemoglobin. I kind of referenced that earlier, that a lot of people don't realize that red blood cells have the ability to transport carbon dioxide. They do, 23% in this fashion. But 70% is going to be transported as bicarbonate ions produced as a result of the combination of carbon dioxide and water because of an enzyme known as carbonic anhydrase, which is found within red blood cells. So we can see our little chemical equation here. We have carbon dioxide and water. This can then shift into carbonic acid, which can then shift into a bicarbonate ion and a hydrogen ion. And we'll really get into this in renal physiology and a little bit later in this chapter when we increase large amounts in the respiratory system. When we increase amounts of carbon dioxide in blood, what happens? How does this affect the bloodstream? And just as a little reference, if we increase the amount of CO2, because of Le Chatelier's principle, if you increase here, the chemicals shift to the right. So we'll increase the amount of hydrogen ions, which will decrease the pH of blood, making blood acidic. But we'll get into that a little bit later. That's a pretty crucial part of Module 7 and Module 8. So hemoglobin. Hemoglobin is just a protein that's going to consist of 4 subunits. Each of those subunits is a single polypeptide called a globin. Each globin is bound to a heme group. Heme is what's giving the red pigment because of the iron atom that's associated with the heme. We have 3 types of hemoglobin. We have embryonic and fetal hemoglobin, and this is going to have a greater attraction for oxygen than adult. Fetal production or fetal hemoglobin production stops after birth, and then we have adult hemoglobin. Now, it's crucial, and we're going to discuss this in class, but I want you to think, why would we need embryonic and fetal hemoglobin rather than just have so-called adult hemoglobin all the time? Why do we need to have a greater attraction for oxygen in the embryonic and fetal stages of hemoglobin than in the adult? Oxyhemoglobin, this is iron and hemoglobin bound to oxygen, so 4 per molecule of hemoglobin. This is what's giving it its bright red color. Deoxyhemoglobin, this is hemoglobin not bound to oxygen, typically is dark red. If you look at the difference between arterial, which is typically oxygenated blood, we'll explain a little caveat of that later, versus venous blood, typically oxygenated blood is far more bright red in color, whereas deoxygenated blood or deoxyhemoglobin blood is dark red to almost purple and bluish, and that's because of the presence or absence of oxygen associated with the hemoglobin. And then we have aminohemoglobin. This is when the carbon dioxide is bound to hemoglobin, and we'll see how that can be an issue. We're going to talk a little bit of how it's transported, but we need to carefully regulate what's binding to the hemoglobin of our red blood cells so that we still are able to move enough oxygen to breathe appropriately or go through ventilation and respiration. So hemoglobin also has the ability to transport nitric oxide produced by the endothelial cells within blood vessel walls, the lining of the blood vessels. And this is going to cause the relaxation of smooth muscle in blood vessels to help decrease blood pressure. Various poisons can affect hemoglobin. Carbon monoxide is the big one if you have a carbon monoxide detector in your house. This is also why they tell you don't stand by your exhaust when you're at the gas station. Carbon monoxide is going to bind very strongly to the iron in the hemoglobin, and this can form carboxyhemoglobin. What this does is that this limits the amount of open space for oxygen to bind because carbon monoxide has a higher affinity for the iron than oxygen does. So if you have a huge presence of carbon monoxide, it will attach more strongly to that hemoglobin, to that iron of that hemoglobin, and won't allow oxygen on, so then you can asphyxiate or have the inability to bind that oxygen, which can cause respiratory distress and even lead up to death. So here we can see a graphic rendering of our hemoglobin. So you can see our different globins here. You can see our heme. So 1 heme for each globin, 4 different subunits. And then you can see in the middle of each heme, we're going to blow that up into a chemical here. You can see our iron located here in the center. So talking about red blood cells and kind of their progression throughout their lifespan, we have to create them, which is erythropoiesis. This is the formation of red blood cells. We talked about hematopoiesis, which is forming the formed elements. Erythropoiesis is strictly the formation of RBCs. And this typically will take 4 days, 3 to 4 days, depending. We kind of talked about the progression of these red blood cells during their production. So we go from the myeloid stem cells to the proerythroblast, to the early erythroblast, intermediate erythroblast, late erythroblast, reticulocyte, which is when it's released into the blood, and then it will mature into an erythrocyte or a red blood cell. Understanding when we're going to have an increase in red blood cells is important because the body has feedback mechanisms to increase or decrease the amount of red blood cells we have. Erythropoietin is a hormone that's going to stimulate red blood cell production. This is produced by the kidneys, and this will occur in response to low oxygen levels. We call this hypoxia. So if you're constantly in a state where you need to have more red blood cells or you're not breathing appropriately, you will stimulate erythropoietin release from the kidneys, and that will lead to the production of more red blood cells, which will increase the amount of red blood cells and ideally increase the amount of red blood cells that can have oxygen bind to the hemoglobin of those and transport it throughout the body. Red blood cells are not eternal. They do have a lifespan, and that's typically 110 to 120 days in circulation. It's different between males and females. It's different between different demographics. But then they'll go through a process in rupture, which is known as hemolysis. So that's the breaking down or cutting of red blood cell. Hemoglobin in the plasma within will then be broken down by different macrophages. And we really do try to recycle everything that is broken down in the body. And this is another example of we'll try to recycle that hemoglobin and those other ions. So looking at red blood cell production here, so we have decreased blood oxygen. That's going to send a signal to the kidney to increase EPO or erythropoietin. That will act on the red bone marrow, which is where we're creating all of our red blood cells. We'll have an increase in red blood cell production, and then we'll have increased blood oxygen. This is the reason that you see many, with the Olympics just ending, this is the reason you see many athletes go train in places with very high altitude, places like Denver, the Mile High City. When you're at a high altitude, you have a decrease in the amount of oxygen available. When that happens, you have an increased production of erythropoietin. So I want you to think about how an increase in erythropoietin, an increase in red blood cells, is advantageous to someone that's going to be participating in the Olympic Games for events that need long endurance, such as marathon runners or sprinters. Why would a higher availability of oxygen be useful for their goal of winning gold? So now talking about hemoglobin breakdown, macrophages are going to take up the hemoglobin released from hemolysis. Like I said, we're trying to recycle everything here. That globin is going to be broken down into amino acids. The heme will then be broken down, and that iron will be released. So the non-iron part of that heme will be converted into biliverdin, then free bilirubin. Free bilirubin is then going to join various acids to form a conjugated bilirubin, which is more water soluble, and that will then be added to bile and pass as feces. And here's just a little loop showing you how we're going to break down that hemoglobin and recycle it and excrete it out as feces. Same here. I don't want you to get bogged down in this. Knowing the general trajectory here on this slide is enough. But I would read through just so that to really bring the point home. So now talking about white blood cells. White blood cells have a nucleus and attract stain in slide preparations when we're looking at them histologically. We'll group them into granulocytes and agranulocytes based on their appearance under a microscope. We already talked about how granulocytes typically have large granules, lobe nuclei, and this will be your eosinophils, neutrophils, and basophils, whereas the agranulocytes doesn't necessarily mean that they don't have granules. They're just not easily as seen under a microscope. And these will be your lymphocytes and monocytes. So here's your standard blood smear. Looking at this underneath of a microscope, and we're going to go over this in class, but I would prepare and discuss with your classmates which each of these is in class. These may appear on a quiz. So talking about characteristics of white blood cells, their whole function is to protect us from invading microorganisms and keep the body clean of any of these foreign invaders or debris and trash that our body has created, whether it's removing cells of ours that have died or debris that these cells have created. There's a couple characteristics that help them perform these functions. Amoeboid movement. This is a directed movement that's similar to an amoeba if you've taken Bio 1. Diapedesis, this is when cells can leave the bloodstream by becoming extremely thin. They kind of squeeze down. They can elongate, become really long, and they can move between or through the endothelial cells of capillary beds because they have to be able to get from the bloodstream into tissues, and they do this via diapedesis. And then chemotaxis, I always thought this was so fascinating. It's the attraction and movement towards a foreign material or damaged cells, accumulation of dead cells and bacteria, will stimulate chemotaxis, which allows those white blood cells to kind of smell it out. They're sensing chemical release, and they will start migrating towards that chemical to then act out and perform their functions. So neutrophils, we kind of talked through all these, but we're going to go through each one once again. Polymorphonuclear neutrophils, PMNs. So these don't stay in circulation very long, 10 to 12 hours, very short lifespan. So they'll stay in circulation, then they'll move into various tissues. They can become mobile. They can phagocytize bacteria. They play a role in the antigen-antibody complexes and in destroying any other foreign matter. And they secrete what's known as lysozyme. This will last for 1 to 2 days. Lysozyme we'll talk about in the next section. And this is the majority of your white blood cells. Sorry, going back to the look. So you can see here that you can see the granules. You can see our multi-lobe nucleus. Eosinophils are pretty readily and easy to see. They have this really granulated and pink, dark pink look. So they'll leave circulation. They're able to enter tissues during an inflammatory response. Huge amounts during allergic reactions. This can cause issues in the respiratory system if you have something like asthma. They're going to destroy inflammatory chemicals such as histamine, and they're going to release chemicals that would destroy parasitic worms such as tapeworms, pinworms, flukes, and hookworms, and they account for 1 to 4% of all of your white blood cells. Basophils, these are the least common type of white blood cell, typically in a stain blue or purple. You can see that it's granulated. They're able to leave circulation. They can migrate through tissues, play a large role in inflammation response, allergic reactions. They produce histamine. Histamine is what is going to increase inflammation. If you've ever taken an antihistamine, you're trying to decrease the amount of inflammation. Histamine increases the amount of inflammation, and also can secrete heparin, which is going to inhibit blood clotting. These account for 0.5 to 1% of all white blood cells. Lymphocytes, these are going to be produced in red bone marrow, but then they'll migrate to lymphatic tissues, and that's really when they're start to proliferate. You can see here that they're pretty easily distinguished because a majority of the nucleus takes up the cytoplasm. They have very little cytoplasm. And they're responsible for antibody production via B cells and direct destruction of viruses, microorganisms, and infected T cells. And these will be 20 to 40% of the white blood cells. We'll learn more about lymphocytes in our lymphatic system and immune system chapter. And the monocytes, these are going to remain in circulation for 3 days. They'll leave circulation and become macrophages, which are just phagocytic cells, means that they're cellular debris-eating cells. They can break down antigens. What's interesting about them is that they can actually take these antigens and go present them to lymphocytes for recognition by those lymphocytes, which is really what the whole lymphatic system chapter will be about. And these will account for 2 to 8% of all white blood cells. Platelets. So platelets are just cell fragments that are broken off of megakaryocytes in the red bone marrow. They have various proteins and sugars on their surface that allow them to adhere to other molecules such as collagen, and we'll see that during the clotting process. Very important in preventing blood loss. They help form platelet plugs, and they promote formation and contraction of clots, which will end up sealing and healing wounds. But they're not very long-lived.